All right, I think we're live. All right, welcome everyone. Hopefully you're here for Workshop 6, Multi-Platform Metabolomics Workflows, Role of Modern Day High-Performance GCMS as a Vital Enhancing and Complementary Tool. So welcome. I guess first of all, I wanted to get the clicker working. Say Happy Father's Day to all the fathers that are here in the audience today. And I had my wife wrangle this picture up. This was quite a few years ago. My oldest daughter in this picture is 21 now, just for reference, and the little one there is just turning 19 in a couple weeks. For those of you who might know Dave Alonso, he's one of the presenters this week. He's one of the nicest guys you'll ever meet. When he saw this picture, he asked, "Who's that young, thin guy in the picture?" And that did happen. He did say that. So we've got a really good list of presenters for everyone today. We're going to start out with Catherine Winder, then we've got Luis Valdiviez, Brian Ruddy, Pierre-Hugues Stefanuto, Dave Alonso, and then Heather Bean. So with that, I'm going to go ahead and turn it over to Catherine.
Thank you. So good afternoon, everyone, and thank you for inviting me to speak this afternoon. Oops, I'm a little. Thank you. Okay, so I've got the quite easy task today of giving you an overview of successful experimental design and QA/QC principles in untargeted metabolomics. So, just to sort of give a quick overview of my presentation, what I want to do is to introduce you to the importance of experimental design in metabolomic studies and to define the key principles that we need to. I'm just not tall enough, am I? That we need to achieve for effective experimental design. And I'll also sort of introduce you to how we embed QA and QC in our untargeted metabolomics workflow. So I'll start off with just an introduction to the approaches that we typically apply in metabolomics. So we've got discovery, and we've got untargeted and semi-targeted. And with this approach, we're trying to maximize the number of metabolites that we're seeing. And with the untargeted approach, we typically do not know the metabolites that are biologically important, relevant to us. On the semi-targeted side, again, we're trying to maximize the number of metabolites that we're detecting. But with this approach, we typically know what we're actually trying to measure. And then, in contrast, you've got the targeted approach. And here you know what metabolites you're interested in. You're typically looking at less than 20. You do a lot of method development and validation. And this is often applied when you're validating your studies or translating them at the end. For this presentation, I'm really going to focus on more the untargeted, semi-targeted approaches that we apply. So, what are we trying to do in untargeted metabolomics? We're trying to measure and annotate as many metabolites as possible. And to do this, we need to apply complementary assays so that we maximize the coverage of the metabolome. And this can include GCMS, LCMS, NMR, for example. So GCMS provides us with a great complementary platform so that we can increase our metabolite coverage. It's sensitive, it's robust, and it has a large linear range. And one of the really sort of key points is that we've got these commercially available libraries that help us to annotate. And for anybody that works in the field will know the pain of trying to identify your metabolites in your studies. And we can also get structural information by the electron impact spectrum. So if I now start to focus on the experimental design, and in general usage, the design of the experiment is an information gathering exercise where variation is present, and this may or may not be under full control of the experimental. And there's two quotes that I like to use here. So The first one, that left to our own devices, we are all too good at picking non-evident patterns in our data. And the second one is that to call in a statistician after the experiment is done may be no more than asking him to perform a post-mortem examination. He may be able to say what the experiment has died of. And I think these are two really sort of key concepts to keep in mind when you're starting to design your experiment. So, if we start to think about experimental design and we're looking at the considerations across the entire metabolomics workflow, so this includes your sample collection, your preparation, how you acquire your data, and we'll talk a lot about that in this workshop, then data processing, annotation, and the final step is the observations and the interpretation. And there's various points that I've included here on the slide that you need to consider. If I just sort of pick out a couple. So you really need to think about your number of samples and your replicates. And this is really important when you're sort of deciding what data analysis strategies that you're going to apply in your study. Because if you don't do that and you don't have the correct level of replication, then you're not going to have enough power in your study. You also need to think about sort of your sample type and how much material you need. And this is really important when you think about your QA/QC, because you don't just need the material to do the sort of biological sample analysis, you may also need it to create your pooled QC, which I'll also discuss. So just sort of digging a little bit deeper into some of these points, your research question will really influence and drive your experimental design. If you think about the diverse range of samples that we apply in the clinical and biological sciences, you really need to think about what's the most appropriate sample type to answer your biological question. So if you're looking at a biomarker study, you might think about urine or plasma analysis, whereas if you're actually interested in some mechanistic information, you might need to use a specific type of tissue, or if you're kind of working in a cell system as a model system, a specific cell. And it's really easy to discover metabolic changes, but what we need to make sure is that they are actually related to your biological question and not due to some confounding factor or some bias that you may have introduced into your study. And quite often, these confounding factors are variables that are not directly sort of studied, and they cause inaccuracies or bias in the measurements or the prediction of the outcome. Now, unfortunately, it's not always possible to control these confounding factors, but what we can do is make sure that we collect sufficient metadata so that at least we can look at this and we can track it, and we can perform appropriate experimental design. And I think one of the key points that I would sort of share with you is really providing enough time in this process and make sure that you plan and review and you seek advice right at the start of your experiments, because ultimately it's all about providing confidence in your results and confidence to you, to your supervisor, to the reviewers of your papers and your grants. So it's really worth spending that time. So I'd now like to move on to quality assurance and quality control in metabolomics and how we embed this in our metabolomics workflow. So I just want to sort of share this with you. So I'm going to start with some definitions here. So assurance is the act of giving confidence, and it's the act of making certain. And quality assurance is the planned and systematic activities that we should apply to ensure quality across a process. In contrast, control is the way to evaluate, to indicate if corrective response is needed. And quality control is the observation of the techniques and activities used to evaluate and report quality. And this involves the measurement of quality across all of the processes. So typically, QA is performed in the sort of planning stage before the data is collected, and the QC processes are performed afterwards. so that you can measure the quality of the data. And these are combined together to do the quality management process and applied in the workflow as part of the QMS. So I've listed a few examples of the quality assurance process here, just to highlight you're all going to be working in a suitable laboratory environment, and you should all be doing training and receiving appropriate education and skill-based training so that you can collect your data efficiently. And as part of that, you should be doing training records, potentially competence tests. These are all examples. On the instrumental side, you'll have suitably maintained and tuned and calibrated instruments. And you'll have lots of documentation which sort of show these approaches and have SOPs. And quite importantly, you should also have procedures for implementing preventative and corrective actions. And these are all examples of the QA process. So if we just think about this a little bit more specifically in the metabolomics workflow, we've got all the SOPs and the processes that you would apply on the experimental side. You will also have the SOPs and the sort of competence statements and training relevant to your data processing and analysis side, because obviously this is important as collecting the data. So we're looking at sort of this process across the entire metabolomics workflow, ultimately to get this fit-for-purpose assay at the end of this. And within your SOP, you really need to indicate where you need to collect certain records. So, for example, batch numbers of chemicals, expiry dates, how long you're going to keep your samples for, how long are they going to be stable, defining all your preparation and processing steps, etc. So now I'll move on to sort of the quality control side of the untargeted metabolomics. And what quality control is doing is providing us with the evidence that the quality requirements have been reached. Now, we can use a variety of types of quality control standard samples to do this. So, for example, we can have the system suitability. We can have different reference samples, and this can be standard. You'll also use internal standards and blinded samples. And a big part of the untargeted metabolomics workflow is we talk a lot about pooled QCs and about preparation blank samples or solvent blank samples. So how do we use these samples in the untargeted metabolomics? So with respect to our standard reference material, you can have an interlab QC sample, and this is a sample that's used within one lab to assess differences between studies. You can also have an interlab QC sample, and this is a sample that we can use across more than one laboratory to assess the different laboratories. You will have a system suitability sample, and this can be something where we're up to sort of 10 metabolites with different RI and mass that are prepared in a matrix-free solution, and you will perform this analysis at the start of your data acquisition so that you can ensure that your instruments are operating to your specific acceptance criteria, and review data such as the RI, the peak shape, the response, etc. So in terms of the pooled QC samples, we can have two different types here. We can have the intra-study QC sample, and this is really what we're often referring to as the pooled QC sample. And it's a sample within one study, and it's created from small aliquots of each sample which are included in your entire biological study. You can also have an inter-study QC sample, and this can be typically applied across multiple studies. It can be one or multiple, composed of multiple sample types that are pulled together. And if you're doing this type of QC sample, you really need to consider the stability of this over time and how much you require so that you have specific for your purpose. So the development of the QC samples in untargeted metabolomics was really quite a pivotal moment, and it started back in 2006. And this was because the drift over time of mass spec really limited the application in large-scale metabolomics studies. And I've just included a couple of examples from this initial work, and these were really sort of the processes that we now routinely apply in our workflow in Liverpool. So how do we apply these QC samples in the pipeline? So first of all, we've got the blank samples. Now this is typically, from my perspective, it's a blank sample without any biological material that I've put through my entire sample preparation step so that I can remove the non-biological chemicals from the data matrix. And then we've got the pooled QC samples. And this is used for various applications, and it's analysed intermittently throughout the analytical run to one, condition or equilibrate the system, and we can see in the plot on the far left-hand side that initially we've got high variation in the signal there. We can use this to perform QC filtering and reporting, so we can apply PCA for a quick and visual inspection, and then we can apply our parameters to do the QC filtering. And then we can use these QC samples to actually measure the drift and do corrective action if necessary. And finally, this allows us to integrate multiple batches of samples into a single data set. So you can see there's various applications here. So in summary, I hope I've demonstrated that effective experimental design should really consider all aspects of the metabolomics workflow. And my take-home message here is when you're developing your plans, really sort of ask and seek advice and plan before you even start your data collection. Now, if you want further information about the sort of workflow and our way of doing these experiments, then please come along to the reproducibility and QA/QC session later on in the week, where David will present some of the work that we've been doing in collaboration with LECO. And finally, for those of you that are new to applying GCMS in metabolomics, I'd like to sort of just highlight that we will be running a training course later on this year. And within this course, it's a face-to-face course, and you will gain hands-on experience of using the instrument. So my contact details there and the training centre Twitter feed. So I hopefully will sort of see some of you in the future. Thank you very much.
All right, I think we do have time for maybe a couple questions. Okay, I guess we'll move on to the next presenter. Thank you, Catherine. So our next presenter is Luis Valdiviez from the University of California, Davis. All yours.
Hello. My name is Luis Valdiviez. I'm the lab manager and GCMS team leader at the West Coast Metabolomics Course Center. So, who am I and what do we do? We started the lab back in 2004 under Oliver Fien at the Genome Center at UC Davis. We've started off with 2 GC-TOFMS back then, and it since increased to 2 more GC-TOF systems and 1 more GC-BT system, all by LECO. And they're still running today almost after 20 years. So it goes to show how robust the LECO systems are, even with just a little bit of maintenance here and there. And I joined the lab 10 years ago, starting as an undergrad and then working my way up to lab manager. And the GC-TOFMS still produces about 45% of our revenue in terms of projects for the center. So I'm here to talk about differences between non-targeted and semi-targeted workflows, and specifically more so in the non-targeted side, because that's what I typically do. And the main differences are you'll see that in non-targeted workflows, as Catherine already stated, that it's more hypothesis-driven, where you don't know exactly what you're looking for. And then you go to targeted workflows to validate, confirm, and see if there's any other pieces of information that might clue in on what's going on in your study. The focus on the non-targeted workflow is going to be everything. You don't know what you're looking for specifically, so you want to capture everything, versus the targeted ones, you're specifically looking for specific analytes. And the coverage, well, depending on the assay and method and how you prep the samples, you can easily get over 1,000 plus metabolites. Majority of them are going to be unknowns, or versus the targeted, where you're only targeting in on those key metabolites. And then for non-targeted, you typically don't need to use internal standards. You can, it helps. But on the targeted side, you definitely want internal standards for correction and other things. And then reporting, usually on the non-targeted side, it's all just peak intensities, peak heights, normalized in some way or form, versus the targeted portions, it's absolute concentrations based off of area ratios. And then the instrumentation portion of it, you can do single quads, you can do the TOFMS, Pegasus TOFMS, the BT for the non-targeted, and then on the targeted side, you can do GC single quads, you can do triple quads on both LC and GC, and then if you're at a curve, you can even do the non-targeted systems like the TOF, and it'll be semi-quantitative because you're not getting that sensitivity as, say, a pure targeted instrument. All right, so starting off, where do we start off? Well, we go ahead and start on sample plasma serum protein precipitation. And now this isn't the only way to sample prep your samples. There's different extraction procedures that you can use when you're working on your study design and ultimately what you're looking for in your samples. We focus on plasma and serum in the core lab, so that's what I'm going to be focusing on here. We start off with 30 microliters of plasma, just a simple protein precipitation using a mixture of ACN IPA water, and we crash out the proteins, grabbing all the metabolites we're interested in. We do a little quick centrifugation, and then we will split up into two portions, into two aliquots, one for analysis, one as backup. Mainly that way, in case something happens the first time, we have that backup sample to reanalyze if something happens. Once we dry it down, we go ahead and resuspend with the 50% ACN. This has helped to remove any phospholipids, anything else that we don't want to see that doesn't volatilize too much in the inlet. And then we dry it down again, and then we derivatize it to that way we can increase the volatility of the compounds we're interested in. And then it gets injected onto the instrument. So why do we do metabolomics on GC? Well, there is a bit of discrepancy between LC and GC and what we can and can't see. On the GC side, we do see the high volatility compounds a lot better. Makes sense, we're trying to go into the gas phase. And we also see the low polarity stuff as well. There is a bit of an overlap between them, but for the most part, this covers a portion of metabolomics that LC can't really pick up. Specifically, you can try doing reverse phase to get all the lipids and other fatty stuff you're interested in, or you can do Hillick to get all the polar stuff and biogenic amines. But the problem is that there's never a good mix of the two. GC is a very complementary thing where you can actually pick up everything on one go, including sugar phosphates, amino acids, some TGs, some pop up that are low enough, but for the most part, it's a good mix and balance between the two. And why use GC? Well, you can get all of this in one injection. If you go over to LC, you can go ahead and change up the mobile phase with one more injection, change the column type, another injection, and different modifiers in your mobile phase will also be another injection. But if you can see them all in GC, just one time and go that route. Pretty straightforward and simple. But not all these compounds turn into a gas. And that's the one problem we have. So what do we do in the GC land? We go ahead and do derivatization. It's a very simple, straightforward. In our lab, we do a two-step derivatization using methoxyamine and MSTFA. And what we do is we're replacing those acidic protons with TMS groups to decrease the volatility of the compounds. That way they can turn into a gas and flow into the GC and through the column easier. Now, how do we do that, and what are the pitfalls? Well, there's different types of derivatization reagents. There's MSTFA, BSTFA, there's another one I'm forgetting off the top of my head, but typically in our lab we use MSTFA, and It's very great for picking up the sugars, sugar phosphates, as well as biogenic amines and almost anything else that has an acidic proton. But one of the issues with it is that we do see different peaks based on the number of TMS groups attached. If you could look at the picture, you'll see with serine, you can get either one, two, either all 4 TMS derivatives, or a 3 TMS, or just a 2 TMS. So you have to know exactly what you're looking for, as you can get multiple peaks in the system. When reporting, we usually just combine the 2 or 3 that we see and report that as the 1 serine peak. But if you choose, say, BS-TFA, a much bigger molecule, tert-butyl group, you'll see the majority being at the 3 TMS, as it won't pick up the second high acidic proton on the amine group. Makes it a little bit easier. So why do we still want to do GC? Well, the separation power is completely there. When it comes to sugar and sugar phosphates, and even sugar alcohols, you get complete separation or close to complete separation on them on the GC. You got a 30-meter column, so you get better resolution power. Versus on the LC, you can try as best as you can, but even in our lab, we're having issues with the sugar alcohols, which is what, if some people are interested in, we say, just do it on GC. It's far easier. Why time of flight? Well, with time of flight, it allows for a higher acquisition speed and better signal deconvolution. And the reason why is because when you're trying to figure out, separate spectra out, you want to make sure you're not mixing spectra with different ions from different compounds. And the TOF allows us to separate those out and create a database easier for comparing across different studies. So we've got the column, we've got the time of flight. Now how do we go ahead and make a database out of this? Well, we standardize it by using retention time and also using internal standards to make a retention index. In our lab specifically, we go ahead and use a series of fatty acid methyl esters, in which case it's spread across the entire run, and we use those as retention index markers for each and across each study and sample that we run it with. But not a lot of people use the PHENE database, which is why we also have a translational tool for the alkane method using cobots, because alkanes are somewhat easier to acquire as a mixture. But recently, we have worked with Sigma in order for them to make our mixture that ways people like yourselves can go ahead and purchase it and use it at home. And then you then use other software that uses our system. So how do we make a database? We have the retention times, we have the spectra that's deconvoluted using time of flight. Well, what we do is we go ahead and take your chromatogram, you go ahead and take out your peak of interest, and you deconvolute the spectra to make sure it's only specifically that peak. We then take that information and save it in what's known as a bin, and this is why it's called bin base. Each bin has a specific retention index and a specific spectra that's going to be consistent and cleaned up. And as we go through and run on other samples, we then reference the same spectra in the bin, which is going to be a specific metabolite, and report that out. Makes it pretty straightforward. It will go through an RI filter to make sure you're in the right areas of what you're interested in, and then it compares the mass spectra. And then if it shares the same M over Z for it, it'll try to look for which is the better match. And once you do that, you can start applying your samples to it, and BinBase will spit out a report. And this is typically what it looks like. On the far left side is where we have all of our annotation and metabolites name, along with any database identifiers, including PEG, entry keys, and PubChem. That way people can use that data and go to network mapping tools and apply it there. There's also, for each sample that we run on the top-hand side, you'll see any of the metadata, organ, species, treatment. That way people can go ahead and classify everything according to their study. We also report everything as peak intensities, and then at the far right, you'll see a pool sample that we run as a QC. And a lot of the times, this is what we present to the clients that come to the core. And just as an example, we ran a mouse tumor cells, and we see a wide variety of metabolites and a wide variety of classes, including amino acids, sugar, sugar phosphates, some lipids in there, some higher lipids, and even lower lipids that we see. So the question is, you're one of our clients, you see this data, how do you know it's true? This is just an Excel sheet. Well, we're in academia, so we have everything open to the public. Welcome to BinVestigate. BinVestigate is a tool that clients with the data set they're provided from us can go ahead and look up every bin in the system. What you do is you go ahead and either type in the bin number or the analyte name in there, or what you can also do is if you want to query something, you can query a species or organ and see what are the top hits from BinBase. And the BinBase is a very powerful tool because every time we've run a study on it for the past 20 years now, we've collected the metadata of the species, organ, and we've calculated out the average peak intensities from them. So if you're interested in alanine, you can go ahead and pull up the spectra, and this is the spectra for alanine, which also includes the retention index for the phene library and the COVATS retention index. That way you can compare it to different studies, including InchiKey and other information which links to MONA. Now, let's say you want to go a little bit further. What else is alanine found in? And BinVestigate allows you to go into it, and you can see in the pie chart, the innermost one is going to be alanine, and the next layer is going to be the species, and the next layer after that is going to be the organ, specifically for that species, which makes it pretty nice to figure out, oh, are they sharing anything between species or not? And will give you the average intensities between them. Well, let's say, well, I still don't trust it. Can I have the raw data? You absolutely can. Besides Chromatoph, which we acquire all of our GC data on, you can use Chromatoph Sync if we give you the right files. Unfortunately, it doesn't work with the old Pegasus systems. But there's other software like MS-Dial that you can go ahead and upload your data and review the spectra and any other information. And the great thing is that MS-Dial already has the phene library incorporated already. So if you already have either the alkanes or the phene FAMEs, you can go ahead and use that for your data processing needs. So you've run your samples. And in this study, we've done 4,000 samples. It's spanned a course of a year and a half, 4,000 samples, a year and a half, roughly 2 instruments. You get a bunch of batch effects. How do you normalize your data? Well, there's different ways to normalize it. You can either do sum normalization, you can do internal standards normalization, but those are not going to work either. So you can try doing surf normalization, systematic error removal using random forest. But in this study, because the batches were just so wide and different, it didn't really help us out. So we ended up using a new one that we generated in the lab called SERTA, Systematic Error Removal Decoding, Denoising Autoencoder. And as you can see here, we have the 4 different samples in the raw data that was run on 4 different time points, and we're able to normalize it and get rid of that technical variance all the way down to less than 20%, which makes it really great because if you look at the bottom right corner, you can see that small differences in treatment groups that are now being separated out from the normalization. And another little application this was done about 10 years ago now, where we're trying to figure out what can we see in foods. So the graduate student at the time decided to do a different plate, a California plate of what is healthy, a Davis plate UC Davis, where the common UC Davis undergrad would go ahead and eat, and then a USA plate. And what she did was she blended it all together. We went ahead and did a volatile analysis using SPEME headspace, and then we also applied our Metabolomics primary metabolomics workflow through the same system. And using the SPEME, we're able to see the differences between the actual plates. You can see in the Davis plate that there's some higher compounds versus the USA plate, and there's differences between the USA plate and the UC Davis plate, which was pretty nice. And the one thing is that because it's untargeted, we still have that data file. We still have all that information. So I was actually talking to her and saying, hey, PFAS is becoming a big thing. Do you want to go back and see if we can find anything? And she goes, yeah, let's do that, but I need someone to do the data for me. And she's like, I'll do it for you. I'll find those PFAS. And with the new NIST library, it's introducing PFAS into their library with the derivatizations. I can go back with that data set, incorporate it, and see if I can find out and dig out any PFAS that were unknowns before. And that's pretty much the untargeted portion of the workflow. So now let's get into a little bit of the semi-targeted. So in semi-targeted, you use it after discovery because you now know what you're looking for. You want to make sure you want to tease out specifically what compounds of interest help benefit your story and describing what's exactly going on. And you do this by selecting several standards, several analytes of interest. You can either do 1, 2, you can do 10, you can do a couple hundred if you really want to. More work means more expensive. And then there's a bunch of different curves you can use alongside for analysis. And then with the addition of internal standards, you can make calculated concentrations more accurate than before. So what type of curves are we working with here? First, you've got the standard and neat external calibration curves where you just make your mixture of your curves and then you run along the side of the samples. But the problem is your samples are in matrix. And you do as best as you can to remove as much of that matrix effect as possible, but you're still not going to get it all out, which is why you can do a matrix external calibration curve. With this one, you go ahead and use like a strip serum or plasma that best accommodates your samples. That way you get those same matrix effects. And this way, your responses will be more similar to what your samples are going to be showing. Sometimes you can't find the strip serum whatever you're doing, so you can go ahead and do a standard addition curve. You make a pool of samples and you go ahead and use that to spike in your curve and then use that for the rest of the study. It's a known, or one sample that's a known concentration. Might be a little easier. But whenever possible, you always want to use internal standards because it just makes everything easier and stronger and less messy. So regarding this, as a part of a core lab, I lost my targeted team. So I went over and did some targeted work, and I was trying to figure out, okay, I've got this study, been sitting in my queue for like roughly three months now, so I need to go ahead and start working on this. It was regarding the tryptophan-curenine pathway. The client came to me saying, hey, where's my data? What's going on? Like, oh, I'll get started on that now. I started working on the LC because that's what we do, is targeted LC, but I was starting to miss out on 2 of my analytes. The last 2, picolinic acid and quinolinic acid, I would run my curve and I don't detect them at all. I see them when I'm doing the MRM transition, but when I run my curve, it's not present. I go to Oliver and he says, well, run it on GC. You can see all of it there. Like, oh, perfect. Yeah, I'll try that. Ran it on GC and I picked up the last 2. One of the things I did not do was, because this was a quick and dirty method, is I didn't try different columns and stuff, and I only tried ESI for ionizing on the LC, but in the core lab, there's not much time to method development as much. And one of the things I was saying is that not every compound ionizes easily on the ESI. So one of the compounds I couldn't really detect, quinolinic acid, as you see here, on the left-hand side is the peak from the LCQ trap, where you see it, mind you, it's very high. It was 5 micrograms on column, and it's a very nasty peak shape. On the GCs, though, 10 times less. It's a big peak overloaded. I wouldn't use that, but when I go down to a lower size, it looks a nice, beautiful peak shape. So clearly, quinolinic acid is better ionized on the EI versus the ESI. And when you're doing semi-targeted work, only use data points that we'll be using in your data set. Over here, we see on the left-hand side 4-ethylenonic acid, where on our higher end, it's completely out of the range, and it's not even consistent between the 2 injections. When you remove those 2 points, you see your curves better fit towards the data set. Now, the only hope is that your samples are actually within that curve. And if it's not, you can go ahead and dilute the samples down to make sure it fits. That way it gives you more accurate representation of what's actually going on in the concentration. And unfortunately, this one, I did not have any internal standards, as this is currently in method development. But with the previous one, I do have internal standards. So in the first curve is without the use of internal standards, where you see that everything's not really linear. And on the right-hand side, you see all of the points are falling more closer on the line with the use of internal standards, as now we're using area ratios. And internal standards are meant to help to remove the matrix effects, or even differences between injections, or even differences between the source and detector, but it's not always going to be exactly the same. So, hopefully we learned that gas chromatography is still an important tool for non-targeted and semi-targeted workflows. Time of flight is a high acquisition speed, allows for better separation of mass spectra for better IDing in databases. And then when you're running semi-targeted analysis, wherever you can, go ahead and use internal standards. It'll make your life easier, less of a headache, and better quality data. Thank you.
All right, I think we have time for a quick question.
I have a question about sample preparation.
Mhm.
There is a way to automatize the process of derivatization. Do you have an automatic process or you do manually?
We do it manually. We tried doing auto derivatization, but this was 4, 5, 6 years ago. We tried doing auto derivatization with the robot arm on the instrument, but our RSTs weren't as consistent as it was done manually. But what we've been switched over to is that we started using pipetters to make it easier for us.
All right, thank you. I was a bit offended by the USA plate. I can't believe you chose cookies instead of cheesecake. All right, Brian is our next presenter from Corteva, Agrisciences. All right, Brian.
Thank you, Joe. Let me see if I can figure out how to work this thing. It's cool. Green is go, I think. All right, so my name is Brian Ruddy. I work for Corteva Agriscience. Today I'm going to be talking about our large-scale GC time-of-flight metabolomics workflow, which sits inside our larger metabolomics workflow. And this is plant-based. I'd like to thank Joe and the LECO crew for the invite, also for making instruments that are kind of easy to work on and robust. So, yeah, so I work in I manage a small molecule analysis lab that is fairly well equipped in both GC and LC instrumentation. We are first and foremost a global reference chemistry lab for oilseed breeding, which is canola, sunflower, and soybean. I like to refer to our metabolomics program as something that we do for fun. And every now and then people take it seriously where we work. But as you can see, we're heavily automated. We have 2 Hamilton robots. We got automated weighers. We have high-resolution Orbitrap instruments, 3 of them. And we have a number of LECO BT time-of-flight instruments. So just a bit of a background, I guess a disclaimer. Here's the marketing materials in the industry talk. Corteva is it's an amalgamation of 3 different companies. Historically, DuPont, Dow, and Pioneer. Pioneer was actually the first commercial breeding organization in the United States, founded in 1920. So pioneers in seed hybridization and breeding, and also actually one of the first data science and informatics companies. Most of our understanding of population statistics come from Pioneer, actually applied. DuPont and Dow merged in 2017, and they spun off the agriculture assets, and we are now Corteva. So where the company is kind of situated, we have a seeds, global breeding and traits organization, and that is in Johnston, Iowa. Johnston's a suburb of Des Moines. The crop protection program, which has a lot of similarities to a big pharmaceutical company, that is located in Indianapolis. Crop protection chemistry, so natural products. If you think about these 2 companies and these 2 sites, there's a lot of metabolomics that's done on both sites, but people refer to metabolomics as different things, right? So you have folks in Indianapolis and the crop protection headquarters doing metabolomics for natural products. They're doing metabolomics for strain engineering to improve crop protection chemistry. In Johnston, our metabolomics is plant metabolomics, and the majority of it it's been going on for 20 years where we've been doing it has been for breeding and prediction. Just another plug, I guess, on a rebranding of a company and a market sector that doesn't have the best reputation in terms of sustainability and care for the environment. That's one of the tenets of the company moving forward, is to have an eye on basically doing the right thing in terms of the environment, green chemistry for production, things of that matter. So in terms of the work in the Johnston, Iowa site, it can basically all be summed up on this slide. So, and I guess this doesn't include the discovery of novel traits, but the majority of the operations that are done on our campus are: an embryo is transformed, it is back-crossed in multiple T events, which produce both null copies and then hemizygous and homozygous copies of a trait. So basically, these are pure genetic plant populations with which we can test traits. Historically, we've done a lot of transgenics, where we didn't exactly know where the trait was going in the genome. It was really important in order to phenotype for off-site effect. Now we're going to CRISPR-Cas editing, so we have a good idea of basically the site and the expression that's gonna occur in a safe way for the plant population. So there's less calls to make. But where metabolomics fits in is basically past T1 event, and this is performed in well, the majority of it's performed as a sampling offshoot in our controlled environments, which is so basically every plant population in these early T1, T2, T3 events are on large conveyor belts and with really heavy automation. And you can see on the photo on the right, every plant's on a conveyor belt and every plant's easily sampled. And this is for PCR, for trait testing. This is for protein quantitation for both ELISA and multiplexed triple quad work, and then for metabolomics as well. So our metabolomics historical archive is pretty immense, particularly with GCMS. Right now, the count for maize leaf punch metabolomics is approaching 400,000. I find this an incredibly impressive data set. And actually, it's kind of an ode to the LECO time-of-flight instrument, the Pegasus 3 and now the BT. We do have a couple proposals out academically, which I want to plug. If anybody is interested in machine learning, artificial intelligence, wants to work with a data set of this magnitude, or knows anyone who would be good at it, trying to kick that around both academically, postdocs. We're looking at things on the table, because this is it's kind of an impressive organismal data set. So, but yeah, you heard a lot about this from Luis's talk. GCMS, LCMS coverage range. We do specifically methoxyamine TMS derivatization. So we're kind of sitting in that red box over there. And as Luis showed, you get more of an expansive molecular weight and boiling point range for LCMS, but the data quality, the data consistency with gas chromatography is pretty much unparalleled. This is the kind of just a background on our sampling procedure. You can see the plants on the conveyor belt. We go 4 leaf punches per tube. They're punched into matrix tubes, liquid nitrogen quenched. We freeze dry them, and then we put them onto the automated wire. From there, these samples, the freeze-dried samples, are extracted with a Hamilton robot, 252 chloroform methanol water. They're all internal standard spiked. And then they're dried down before derivatization and run on the LECO time-of-flight instrument. Unlike Luis, we have found use for the PAL autosampler that derivatizes on the autosampler. And I think that's probably maybe just because at a company the labor is more expensive. So I guess we'll eat the cost. And I guess with the ability to oversampling and the fact that we're doing the same samples, kind of live with the variability. But we haven't tested it really. But I do want to make the disclaimer that we do methoxyamine derivatize by hand. So this is just the MSTFA that's loaded by the PAL Okay, went too far. So this is a data rendering in 2D. By the way, I have no idea about time, so somebody cut me off. I'm going too slow. Nice. Data in 2D. This is your classical total ion current against time. We COVATS retention index every batch of samples, and we have going back 20 years. This is actually done by our in-house Metabolomics. It was the software built by somebody who doesn't work at the company anymore, but it still works for us. So we convert the PEG files to CSVs. And this is a look at the data in 3 dimensions with your ion abundance going out of the page and your M over Z and the Y on the last slide. But I put this figure together just to kind of explain to folks on my end who are plant biologists, molecular biologists, the people that I work for, why retention indexing is so important. And it has a lot to do with grid alignment. And for us, hopefully for this informatics project that we got on our hands going back decades, this probably will enable somebody to align a file that was collected 15 years ago to a file that was collected last week. So, but yeah, data is alignable across decades of data collection. So, the idea is to go from these 3-dimensional data files into ideally a spreadsheet, 3-dimensional to 2-dimensional data reduction. And this actually is not just for detection or statistics or whatever. It's practicality in terms of data reduction. But a lot of the experiments that we get have tons of metadata, and we also make use of multivariate statistics, and we're a big user of PLS Toolbox. So kind of an outline of the workflow on the top, GC-TOFMS mass spec collection, and in a lot of the projects that we're working on, people are looking for trait-specific small molecule markers. And there's different ways to do this, volcano plots. There's some nice things about parcel least squares discriminant analysis, whereby you can make a model and you can actually validate for subsequent sample sets that come in. And I kind of want to harp on this, which is really important in whether you're operating an instrument and you're direct infusion where you're collecting scans, or whether you're doing a metabolomics experiment where you have tissue punches from 2,000 plant samples, right? So your ability to data mine in a really, really well-oiled, high-quality system is proportional to 1 over the square root of the number of samples. That's classical signal averaging. So basically, the practical take-home message of that is you're going to double your signal for 4 times the number of samples introduced. So I guess with that being said, this is a typical experiment that we get in-house, where we are doing a number of users are doing a number of typical experiments under a number of different conditions, experimentally, environmentally, tissue types, growth stages, and then on the right you can see pedigrees are coded. Those are genetic lines where somebody put a trait in. And basically this entire experiment is just 1 trait and 1 control at a variety of different conditions. And this is one of the reasons why we use partial least squares discriminant analysis, because it kind of helps you sort through a lot of this complex metadata. For this particular experiment, you can see on the left we have this entire data set colored by plant tissue type, and then on the right by gene or control. And you can see I would refer to this data set as most trait metabolomics experiments as a noisy system with regards to the trait. So, your markers are a needle in the haystack. PC1, PC2, you don't see a huge difference in trait versus your control. Going further, coloring this all different ways, it's, same story. Trait versus control is usually the small source of variation. But this is one of the advantages of partial least squares modeling. And you can see on the top, you got your sensitivity. In the center, you have your regression vector, I think, which is your direction, upregulated or downregulated relative to whatever you define as your trait or your control. And then the bottom is your specificity. And this is actually a penalty, basically based upon covariance. So if you have a marker that's really robust, regardless of growth stage, tissue type, whatever, it's going to have a high sensitivity score, or it's going to have a high specificity score in addition to that high sensitivity score. And that's kind of shown right here. You can see our growth stage up in the top left of these figures. That's V. These numbers mean something to you if you know a lot about corn. But this particular marker is heavily growth stage dependent, so it's got a really low specificity. This happened to be beta-tyrosine. Marker really wasn't important here. It's identity. We happen to identify it. It was in Golden Library. It wasn't in NIST. But this proved to be a pretty good marker for phenotyping for inbred populations, not hybrids. We have accurate mass for absolute determination of this with by standard. Make sure that it's the compound that you want. But this is just kind of one illustration of how you can use metabolomics or untargeted mass spec in the GC world in order to get one thing that's useful. And I know there's a lot of stuff on systems biology that you're going to hear today where you can do a ton of different things with metabolomics, but sometimes you can do something really simple too. So in conclusion, laboratory throughput, huge. We love the LECO Pegasus BT, obviously on campus where we are. Alignment from alkane retention index, historically is a good practice, especially if you're doing the same tissue, for years and years. Partially squares discriminant analysis is something that we like if you have really complicated metadata. Sampling power is huge. Obviously things harped upon by Catherine and specificity an important metric when combining data sets for increased sampling power. So with that, I'd like to thank everybody. Sorry for going over time. Particularly Jan Hosbrook, who was actually the manager of this lab before me, Teresa Harp, who worked here for 24 years, who just retired. Also Joe Shambo from Gene Data. Gene Data it's an incredible program if you're chugging through a ton of different files. So I will take any questions if you have them.
All right, thank you. Thanks, Brian. Questions for Brian? All right.
Since you have decades of data collected on now more than one system, are you expecting to see system-specific biases in your data sets that also need to be dealt with while you try to have all the data talk to each other?
Yeah, it's a really good question, right? Because I think the breakdown is, I think it's about 310,000 files on the Pegasus 3 versus the BT, and the BT's got the brighter source. We have in-house processed 100,000 of these files. We've actually grid aligned 100,000. We took all the ones that we have growth stage metadata on, actually prepared a poster for ASMS like 3 years on this. And the alignment is pretty darn good, but the sensitivity it's an issue because it's an order of magnitude scale. So obviously something that we're looking for from somebody who's pretty good with informatics and doesn't mind digging into this. But it's a great question. Thanks. Yes.
Hi. Oh, sorry. Yeah, great talk. A very practical question. You mentioned that you were using your PAL system only for the MSTFA step. Is that just because it's not set up for the previous steps, or that you actually found that it wasn't as good as a human?
It's a good question. Actually, can you ask it one more time?
We're all tired. I was asking whether you're using your PAL for MSTFA only because that's purely technical, that it can't do the other steps, or that you
Oh,
actually
the
find that the human is better.
Yeah, the methoxyamine derivatization. I'm not sure we can get a third diluter tool for the PAL autosampler. I inherited this lab. It's just the way it's always been done, and that they previously did this on the Gerstle, on the Pegasus 3s. So I'm not 100% sure. Can one of the LECO guys take this? Can you get a third diluter tool on a PAL autosampler?
We haven't tested a third diluter tool, but we can perform both methoxamation and silylation with a two-tool unit. Smaller 10 microliter syringe for injections, and then a larger 100.
2 blocks though, right? Because it's 30 degrees, and then the MSTFA
Yeah, we
is...
got two agitators at different temperatures. If
Thanks for that question.
We could describe it later.
Brian, thanks very much for the talk. So how many metabolites can you confidently annotate now, given the history that you've had on this particular set of plants and things?
You know, it's funny, with the majority of the work that we have done historically, you know, why do we have this many sample numbers? Like, it's been mostly drought prediction modeling. So you can actually use, and this would be a really cool talk to deliver. I think Jan has given it potentially previously. But it's probably not any more, if you want to get really good with annotation, I think you want to go to Davis. No, I mean, we're still using NIST. We're using a modified Fiend library. We're using GOLM. I wouldn't say with all of the work that we've done in metabolomics, we're probably any better at annotating than any of the other top metabolomics labs, which is sad. It may be embarrassing. But it's a metabolomics conference, right? No use not being honest.
Thanks, Brian.
All right, in the interest of time, we'll do one more question.
That's a really interesting talk. Since you really have a huge observation pool, and like even though when you do the PCA1 versus PCA2, there's no really clear separation. I'm just wondering, have you ever tried any like a little bit more modern kind of clustering methods, such as t-SNE or PICO-A or like DB scan, etc.? Because the underlying distance measurement is actually more flexible, I guess. Yeah.
I'm not sure I understood that. Can you say that again?
Okay, so well,
maybe speak closer.
Okay.
Or maybe we'll catch up outside. Definitely interested in hearing that question.
So, like, since you only do a PCA versus PCA1 versus PCA2.
Oh, PCA1, PCA2, right?
And you said that there's no really clear separation.
Right. Yeah.
And, yeah. Since you really have a huge observation pool, I'm just wondering, have you ever tried any like a little bit more modern kind of clustering methods, such as t-SNE or PICO-A or DB scan, etc.?
Like if I was to do a k-nearest neighbors or something, some other classification method.
Yes.
You would definitely see separation. But I made those PCA plots just as an illustration to say that, 90 to 95% of the variance right off the bat is not based upon the trait control relationship. But you're right, there are methods that where you could, right, that you could look at the finer discrepancies, right?
Thank you.
Yeah, you're welcome. Thank you.
All right, thanks, Brian. All right, Pierre-Hugues Stefanuto.
Okay, thank you, Joe. So, good afternoon, everyone. So, today I will continue in this use of gas chromatography for metabolomics, but we will move to also like multidimensional GC and see what's the added value that we can bring with this extra dimension. So, I have a really long title that it's almost an abstract there. So when I see that, I was like, okay, let's use it to my advantage and like follow the presentation with it. So if I focus on the first part, so comprehensive two-dimensional GC and how it's enriching the number of species detected. So for that, I was like, okay, maybe not everyone here is familiar with GC-GC, so let's step back and discuss a bit about the technique and how it's based. So I'm from Belgium, so when people ask me how I do GC-GC, I have a beer-based example. So I think it's almost beer time, so I think we can go with that. So imagine that your sample, your complex sample, it's a mix of beer caps. So here, all the molecules that you have are beer caps, and here it's all the Trappist beer that you can find in Europe. So when we do GC, what we do, basically, we choose one criteria of separation, and we apply it to the mixture, and we organize the molecule, or here the big caps, based on these parameters. So here, if I choose the Abbey, I see that I can see how many Abbey are producing Trappist beer, but still, we see that more than half of the Abbey are producing more than one beer. So we have more information, but not all the information there. So we can try to push the method, but if we go for long separation, long colon, we will have some technical limitations such as peak broadening, run duration will be long, it's not something that you want, so we need to find another way. Another way is to apply a second criteria of separation. So here, for example, I can choose the beer type, and usually one Abbey produce like different beer type, but only one beer in each type, and you can see the full resolution. So going back to the chemistry. So in the previous talk, we saw that in GC, mostly the separation that you see, it's based on the volatility, so the boiling point. Here, since I can add a second dimension, I can add a second physicochemical criteria, and usually we use the polarity. So now the compounds that have the same volatility but a different polarity can be separated just based on the chromatographic dimension. So this is for the GC side of it. And in my title, we were also speaking about annotation and identification. So how we can use GC-GC for that, and what's the added value of the technique. So because we have these multiple physicochemical parameters for the separation, we can use that as an information. So here I draw color area on my chromatogram. So if you see the red area at the bottom, it's basically compounds that cover a wide range of volatility, but that have a low polarity. So that's basically hydrocarbon compounds. And you have all your hydrocarbon compounds and different carbon number across the x-axis. If you go a bit higher on the y-axis, you have semipolar compounds. Usually it's compounds with one or two heteroatoms, like alcohol, amine, ketones, that you separated at. And on the top of that, you have compounds, again, across the volatility range, but that are even more polar, like, for example, the aromatics. So this is basically the main added value of GC-GC, is this structure of separation. So based on the position of the peak, you already have a good information about the chemistry of the compounds. And you can go even more deeper on that. So if you do plant metabolomics, we were just speaking about that before. If you zoom in the semipolar compounds, you will see on the volatility, one side will be the terpenoids, and after you will have the sesquiterpenoids that are grouped together, and you can have slightly different polarity. So you can really have a high resolution of detail based on the separation. So now we can have the mass spec kick-ins. So like GC, we can have MS detection, and you can also have high resolution detection on the mass spec side. So you can have really a lot of information on the identification. And if you have still compounds that coelute, you can apply deconvolution. So if I summarize the detection and annotation power of GC-GC, we have the electron ionization fragmentogram that is highly specific. So we discuss a lot about libraries. We have the high resolution, so we can have accurate mass determination. We can apply the linear retention index, and we have this pattern of separation that give us information about the chemistry. So when you have small molecules that are GC compatible and all this information, you can have a high confidence on the identification because the probability to be wrong or having two molecules that have the same property is really small. So that's why for some of the molecules that you have, you can reach level 2 MSI identification without any standard injection. So I was mentioning the mass spec, so just I wanted to push in for the non-GC user. So EI is clearly the strength of GCMS because we have all the library available. The EI, it's highly specific, it's analyzer independent. We have the linear retention index, the accurate mass. So all this information are in most of the libraries, but it's also a challenge for the user because we know that it's powerful, so we want to believe it, but sometimes it can get wrong. So for example, if I take the paraffin and the isoparaffin, when you reach about like C15 or C17, you see that the fragmentation is always the same. So you lose this specificity, and you have to rely on the GC or the GC-GC separation to get the information. So it's really important to, if you are interested about this larger molecule, to have your GC side right or to change ionization method, and to also wonder how much is the probability to be wrong. So now, commercial software gives you the probability score, so I will come back to that in my example after, that really gives you an idea of, okay, you have a good match, but how many good match do I have in my library? So what's the chance that I have to be wrong? So last part of my presentation will be really where the fun is. It's example of non-target and semi-targeted analysis where matrix complexity is high and identification confidence can be aided. So the work that I'm presenting here is the work of Kinji Albat, who is a PhD student in the lab. And if you want the full story about the data that I will present you, she has a poster. It's easy, like it's 4x1. And she also just get the result published, so don't hesitate to check her paper. So a little bit of context. This project was based on serum analysis for colorectal cancer metabolomics. So it's just to get fundamental information about what are the changes in serum sample for different type of colorectal cancer and also different stages. So our goal as analytical chemists was to develop a minimally invasive sampling techniques and data sampling protocol and derivatization for this type. And when I say minimally invasive, it's not really for the patient that I'm speaking, but for the clinician. Because unfortunately, when people have cancer, they have blood withdrawing all the time. So they don't mind to give you an extra tubes or 2 extra tubes. But when the clinicians are biobanking this sample for clinical research, they are willing to share with you a bit of sample, but not too much, because they want to still be capable to get this biobank. So we need small volume, not for the patient, but really for this biobanking dimension. So we need small volume. So in this study, we use what we call a more global metabolomics sample treatment. So Luis did a really good job presenting that because it's MSTFA-based method. So we call it global because we see almost everything that is there. We see sugar, fatty acids, all the main amino acids, so everything is there. And we wanted to compare it to a more semi-targeted method where we want to focus on one chemical family. Because when you go global, even with GC-GC, there are a lot and there is a bit too much. So maybe you miss some information because of the amount of information that you have. And also when you focus on a specific chemical family, you also have more higher extraction efficiency on the method. And after, for the analysis, so we use GC-GC, we run the big batch of sample on a low resolution system, and we also replicate some injection in high resolution. So we cannot run everything in iRes because the iRes, you have the gain in identification, but you have a loss in sensitivity, and you have an increase of data complexity. Thus, we use a complementary approach. And it's where you can see really the complementarity of the GC-GC. So this is the same sample injected on two systems. So it's two different mass spec with two different GC on it, but they use the same GC-GC setup. And you can see that you can do translation based on the image. So if you have a peak on the middle top of the chromatogram on the low res, you can check the peak on the medium top on the high res and see that you have both indole, you have fragmentation that is similar, but for the high res, you have the accurate mass on the molecular ion that gives you some confidence in the identification. So we apply that, and today I will discuss mostly about the semi-targeted, so the lipidomics approach. So this is the protocol that we use. So we use 25 microliter of serum, and we have like a two-step incubation and a liquid-liquid extraction. The method was validated by Kinjal, so this is the paper with the validation, and you see that you can use it for all type of samples. So we use it for cancer serum, we use it for pig serum, for food technology development, so it's really a broad application for that. And even when you focus on one specific chemical family, you can also focus your separation power on that. So here we have the fatty acids, and you see that on the volatility range, so the x-axis, you have the different carbon numbers with different type of fatty acids. And if you zoom in and you look to the polarity axis, you can see that you have the number of insaturation changing the polarity of your molecule. So again, even before mass spec identification or any type of screening, we have a really good idea of which type of compound we can have there. So it's really informative. So we did some alignment. So we discussed alignment in the previous talk, and we did some unsupervised screening and feature selection to gain information about the metabolic pathway of colorectal cancer in serum sample. So jumping to the end of the story, like this study allows us to detect 8 features, and these 8 features were highly different in the different type of samples. So we had adenocarcinoma, so a more aggressive type of colorectal cancer. We had adenoma, which was a more benign type of cancer, and we have also control in individuals. And we can see that on the fatty acid approach, we have down-regulation of omega-3 in the adenocarcinoma and upregulation of omega-6, so in inflammation marker. So we have a higher inflammation in people with a more aggressive cancer. This is what we expect, but it was really interesting for us to see that on a proof of concept level, on this small volume, we were able to see that. And when it comes to annotating these 8 features, we can really combine all this information. So we had the structure of the separation, we have still the similarity, the reverse, this probability index that is really important. And you can see on fatty acids, the probability is not crazy. It's low. It's just because you have this long chain of hydrocarbons attached to the carboxylic group, and you never exactly know how many C you have there. But because we have the GC-GC information, we know exactly the C number and the number of insaturation on the molecule that provide us information. When you can get the molecular ion, you can have the mass accuracy and all the figure of merit from the model building. And so just to finish on the visualization of the result, we can see that on the selected feature, on all the features, you can see a difference between the group. But with this top 8 feature for the adenocarcinoma group, we were even able to distinguish the stages of the cancer. So this was not a study designed for that, but it's just that we saw the group and checking the metadata. This is what came out of it. So it was really highly informative to link this level of inflammation to the stage of cancer, because for the clinician, that was not something that they were targeting, but it's a new target for them for a future study. So I want to thank the research group, all the collaborators at the university hospital, and all the the support in the research. And if you are interested about another workshop focused more on the techniques, we are hosting a multidimensional chromatography workshop in California in January, and you are all welcome to come to attend because it's free. Thank you.
All right, we have time for maybe one question.
Thanks, great talk. This is actually a question for all of the speakers today, and that's helium versus hydrogen. Have any of you actually tried hydrogen, and what are your thoughts on it, particularly in terms of changing methods to deal with the change?
So yeah, this is a big topic, and there are more and more interest in changing. We did not directly try it in the lab, but I will pass the ball to LECO because they are doing a lot of research on the impact of hydrogen on the type of molecule that you see, the impact on the fragmentation in the source. So they have a lot of information about that.
Yeah, so we have been doing a lot of work on hydrogen, and we can confirm that both the benchtop unit and the high-res system are capable of running hydrogen. To talk to your method translators, we're using a tool from ResTech. They have a good method translator from going between helium and hydrogen. I don't know that we've done a lot of work in the metabolomics space yet on comparison of spectra, but everything that we have done, the classes of compounds that are typically problematic, some pesticides and things like that, we've seen really good results with spectra, similarities between the two. All right, let's go to our next speaker, and that's Dave Alonso.
Oh, there. All right, I'm just checking to see if I can see that. I didn't bring my regular glasses. I'll have to go from memory. All right, so yeah, if I had a mic. Do you have a mic? Yeah, sorry about that. All I have is readers. Thank you very much. All right, so the talk today is utilizing GC-by-GC with high-resolution MS for discovery workflows. And just an outline of the presentation where we'll talk about instrumentation, the objectives of this proof of concept exercise. I already sort of answered this question, why GC-by-GC? I think they've talked about that. But it's the combination of enhanced chromatographic resolution with high-resolution time-of-flight mass spec, that combination that makes this exercise very, very powerful. We'll go over the workflow because it is metabolomics and it is important, and we do use GC, so there is some derivatization. And then we'll jump into the results and the discussion. Metabolite annotation, unknown characterization, and statistical processing, and then end with a short summary. So we use two different instruments. The workhorse is the instrument on the left, which is a LECO Pegasus BT 4D, but the talk today is going to focus on the high-resolution time-of-flight mass spectrometer. And the objectives, again, of this preliminary work is to demonstrate the utility of the GC-by-GC high-resolution system and modern chromatographic software for confident metabolite annotation. And we want to differentiate biofluids, specifically plasma and urine, using some relatively new statistical software, Chromatoph tile. And the samples were actually prepared. The experiment was designed by collaborators at the University of Liverpool, Rick Dunn and Kate, who spoke earlier today. And so we received the samples. We transferred them to GC vials. After drying, two-step derivatization, it's important to keep the materials dry because the reagent, specifically the silylation reagent, is reactive with water. So we did a two-step drying procedure, speed vac, and then lyophilized at low temperature and pressure. And then we performed a two-step derivatization. I did have a video, but I was told it wasn't going to work. But if you're interested, and there was a question earlier about that, yeah, we have a rail that does both steps. And it is a product from LECO. And then we use Chromatoph to control the autosampler, the gas chromatograph, and the mass spectrometer. There were a variety of samples, urine, plasma, and we had some alkane standards, as well as some quality control samples and blanks. And during the derivatization process, we also injected injection standard, OFN. And then we randomized the order, and we performed just-in-time derivatization to try to reduce experimental variability. And then we used the same software package, of course, to perform untargeted peak find, which also has deconvolution. And we use NIS-20 and Wiley-12, very large databases, well established. And then we did retention index filtering. This is all done automatically, and mass delta and mass accuracy calculations specifically to increase the confidence in the annotation. And finally, we perform downstream statistical processing to try to differentiate two types of samples, spiked and unspiked. And they were spiked, the spiked samples were spiked with amino acids and some other acids. And for those of you who are not familiar, I think that PH did a nice job, the previous speaker, but the typical GC experiment is you use one column and you reach the detector. The difference here is that you have a modulator. I call it a pacemaker, but it basically cryogenically focuses and then quickly injects whatever it focuses, that element, into the secondary column that has an orthogonal mode of separation. So there's a change in the intermolecular interactions between the stationary phase and whatever's going through the column. So that's a typical GC-TIC, let's say, if you have a mass spec attached to it. And then once it goes through the second column, you have a different separation, and you get now a map of your sample. Sort of that old experiment, if you remember organic chemistry, I don't know if you did it, where you stick a TLC plate, develop it, and then rotate it, use a different solvent, and develop it again. You just get better separation. So what does the data look like? Well, it's a contour plot, it's a map. And if you look at the x-axis, in this case we're using a non-polar column, that's first dimension separation. Note the Y scale, it's a much shorter scale, and that's a separation using a more polar column, so different types of interactions between the eluent and the stationary phase. And I can tell you with pinpoint accuracy where some of the compounds are at, because you have what is called group clustering. Something nice that happens is that you remove a lot of interferences that you wouldn't remove in your typical 1D experiment. So the bad side of this contour plot, or as I like to call it, a map, this is the bad part of town. This is where you have your column bleed. There's some dimethylated cyclosiloxanes here. There's a nice little river of alkanes, a little bit difficult to see because I'm zoomed out, but there are thousands of peaks in that map. And we remove the interferences. Of course, that's going to improve the signal to noise, improve database comparisons. But why do we do this? Well, ultimately is because we annotate more metabolites. That's a conservative figure. It says 2 times. It's typically more like 3 or 4 times more compounds annotated. And let me go back one. One of the reasons why you get more, I've selected, there's a whole bunch of these coelutions in the first dimension, but they're separated in the second dimension. So I have 2 sets of compounds, and I'm going to show you the spectra for those 2, or at least 1 set, in just a second. But I've listed after automated processing. The first column has your metabolites. The second column, of course, has formulas, retention times, there's spectral similarity values, and since we're dealing with high-resolution accurate mass, you have also mass accuracy calculations. And that column, that last column, is specifically for the molecular ion, if it's present, all right? I'll come back to that because it really doesn't have to be present, and I'm going to show you why. There's acids, diacids, amino acids, fatty acids, monosaccharides, reduced sugar, sugar phosphates. There's more. There's sterols. There's monoacylglycerides. There's a wide variety of compounds that you never thought you could do by GC. So this is just a representative list. Automated data-based comparison results in an average spectral similarity of 836 out of 1,000. Of course, the closer you get to 1,000, the better. I always like to look at the rich data, right, that's underneath those tables, the results that make up that map. On the top, I have the observed data, and you can see that indeed that it's high-resolution accurate mass data. I have the amino acid aspartic acid. At the bottom, the first hit after automated processing, I have the score of 809 out of 1,000. That's respectable. But we don't rely just on spectral similarity searches. It's not just database matching. We also do mass accuracy calculations on the fly. And mass accuracy is defined there. We are at 1 part per million on average or better. So increased confidence for compound annotation. And that's what PH was talking about earlier. There's a bit of confidence even without without a standard. Another amino acid, methionine, you can look at the top. The molecular ion is labeled. Mass accuracy a little bit higher than we want, but still respectable, minus 1.58 part per million. Spectral similarity is okay. It's at 748 out of 1,000. And we can also look at mass accuracy of any of the ions in that top deconvoluted data. You can see for this derivative, or this 2-hydroxyibuprofen in this case, so a metabolite of that over-the-counter drug, you can see a good spectral similarity value of 931 out of 1,000. But indeed, a lot of times you don't have a molecular ion. That's why there's a question mark there. It would occur around 366, but it doesn't matter because I can calculate, or the software will calculate the mass accuracy for every single ion in the mass spectra. And we can walk back and we can characterize molecules based on fragments. I've selected 3 there. And of course, you can get mass accuracies as well as, I'm going to talk about this in a second, isotopic fidelity for any of those molecular ion clusters. So again, increased confidence in metabolite identification. Another one, tyrosine. Again, no molecular ion. You can see that in the observed. I've identified the base peak given a structure and also a minor peak as well. No molecular ion present, no big deal. Again, 2 fragments that I'm highlighting, mass accuracies of minus 0.15 and 0.30. But there's more, because with a high-resolution TOF instrument, you typically have a source that will provide you molecular ions or adducts. And so with valine, we have the same problem, or I'm sorry, glycine. 3-TMS. Good spectral similarity. Most people would move on with that value with some confidence level. There's no molecular ion present, but we can quickly perform a software switch and collect the complementary positive chemical ionization that's shown there to the right. And you can see that now, where you didn't have a molecular ion, now you have a protonated molecular ion. And furthermore, the software will automatically calculate a theoretical value for that ion cluster. And the comparison between the two is the value that I've have there for isotopic fidelity, which happens to be very good at 994 out of 1,000. But there's even more confidence, right? You could talk in LC world about different features. Well, those are sort of built into the experiment here, into the data file, right? Because not only do I have the protonated molecular ion, I do have fragments, and I can tie those fragments, by the way, to the EI data, if you look carefully. In addition to that, there's other adducts in that positive chemical ionization spectrum on the right side. There's an M plus C2H5 or M plus 29, and there's an M plus C3H5 that's common with that particular reagent gas that you use for the chemistry, right? And if you look carefully at the mass accuracy in the table down below, I don't know, I'm feeling pretty confident in that formula and that identification. So again, confident metabolite annotation. I gotta be honest with you, there's a lot of unknowns. And it's hard to believe with these large databases has been around for decades and continue to grow. I think NIST just released NIST 23, I believe. I didn't search against that particular database. I'm still using NIST 20. I'll test 23 shortly. I set the threshold very low for the database comparison. I lowered it way down, still no match. It's not a problem, right? If this is important, if this particular compound is important, let's say it differentiates my samples, I can calculate a formula for that particular species. I have a little question mark because I'm not sure that that's the formula. We can discuss that offline. But again, I can perform a little switch, and now I'm confident because in CI mode, I get a protonated molecular ion basically telling me that I was correct. And again, I have additional data. That's kind of getting boring now. M plus 29, M plus 41, good isotopic fidelity. I'm pretty confident. I have a good formula. I still don't have a structure. That's the ultimate. So within the software, you can launch a ChemSpider search. There's also a link to an EPA tox library. And unfortunately, it didn't hit like 10. It hit like, what is that, 300 some? But at least I know, get an idea of the elements of unsaturation, but which, by the way, are provided in a tabular form. But I'm more of a structural guy, and that helps me quite a bit. And hopefully I can do a little bit more investigation. So I have this powerful system that gives me this rich, comprehensive data. I have a very easy mode of identification with, annotation. I was told I couldn't use identification. But it's very, very powerful, right? And I need to find a statistical package that can handle this rich, high-quality data. Well, it turns out that we've put out a product called Chromatoph Tile for downstream statistical analysis, And this is based on work by Professor Sinovac at the University of Washington. And it's based on Fisher ratios, or the class-to-class variation divided by the within-class variation. And here are some of the results. I'm going to step to this side because I think I've been ignoring people on this side. It's a wonderful piece of software. So you have your features. I'm going to go ahead and call them compounds after processing. There's a bunch of information that I've taken out of this feature table or compound table. There's a heat map included, and as I select each one of these potential compounds that differentiate the samples, and I've picked in this case an amino acid, L-methionine, and I have a library search. The top is our data, the bottom is the library match. I have a structure. In the middle of a bar chart showing the unspiked and spiked relative quantities, or if you make concentrations. The software generates PCAs. You have the ability to scale and so forth. I believe I'm using mean-centered, and I think I'm using a logarithmic scale in this particular case. But clearly an easy way to go and identify your samples. I picked an amino acid and now a fatty acid, stearic acid, which also is elevated, so probably spiked into the spiked samples versus the unspiked. Again, on the upper left-hand corner, we have the library results. Features are on the bottom, right? Heat map, and the same PCA. And I've listed just some of them. You can filter also. I wanted to have similarity values that were decent. So 777 out of 1,000, and F ratios that were greater or equal to 98. The greater the Fisher ratio, right? The more likely it's a candidate that tells you that you're differentiating the samples. And indeed, I have a whole list of different amino acids. There's reduced sugars, there's disaccharides, there's nitrogen-containing acids, and so forth. And with that, I'm going to summarize and just say that we developed in this preliminary study a way to analyze plasma and urine samples. There has to be some care in how you treat the samples. The extraction, there's many procedures for that. Drying is critical in this particular process. We also performed automated derivatization. In this case, it's a two-step derivatization. We could have done a one-step and put dimethyl T-butyl type silo groups on there. And we use GC-by-GC because we want to see or annotate more stuff. We do easily go between EI and CI, again, for increased confidence in metabolite identification. And of course, we have a powerful statistical tool that can handle this data. And with that, thank you very much for your attention. Oh, a shameless plug. I think Kate already did that. We have a session describing the project a little bit, or giving a little bit more detail. And again, thank you.
All right, in the interest of time, I'm going to go ahead and just let you know to see Dave at his talk on Wednesday. And I'm going to go ahead and turn it over to Heather, our last presenter. Thank you, Dave.
No problem. Oh, hello, I'm on. Okay. Hi, I'm Heather Bean. I'm from Arizona State University, and I'm rounding out the session to talk about our approaches to data analysis, the challenges that we encounter, and some of the approaches that we use. In my lab at Arizona State University, we do microbial metabolomics work and breath-based diagnostics, and we focus on the volatile metabolome, and we use comprehensive two-dimensional gas chromatography to do that work. And often the case is that we are dealing with hundreds of samples, and once we analyze those by two-dimensional gas chromatography and clean up the data tables a bit, we're often dealing with thousands of peaks that we want to now make sense of. And we're often taking two different tax, sometimes we're interested in biomarkers. We want to know what is predictive and different between Class A and Class B of our samples. And we'll use machine learning techniques or multivariate approaches like random forest, PLSDA, and so on. But sometimes we're interested in global patterns across the entire data set. We're not just interested in what's different between two classes, but we're trying to describe an entire class of samples more holistically. And we're often using methods to visualize that data, such as principal components analysis, hierarchical clustering, and so on. I want to emphasize, and the term alignment was brought up a couple times earlier, that data alignment is required before you do either of these processes. So let me just talk briefly about what data alignment is and why it's necessary. Alignment's required when you want to compare samples, and it's for two reasons. Brian talked about how you want to index or match up the retention times of samples across chromatograms, and that's one really important step and component of data alignment, where you have two peak tables from two different samples, but the first hit in the peak table, the first compound detected in peak table for sample 1 may not show up until the third in the list for the peak table of sample 2. And so alignment will look at retention times and mass spectral similarities between those samples to make sure that we're comparing apples to apples and oranges to oranges across the two samples, the two sample tables. But in addition to that, alignment is also ensuring that you are measuring the peak intensities or reporting the peak intensities the same way across all of your samples. So it may be that you're just using TIC, and that's much more applicable across many different samples. But what we are often doing in our studies, and what is the default mode for the software that we use from LECO, which is ChromaTOF, is that alignment is performed on unique mass. And the unique mass will be a mass within that mass spectrum that has very little interference across all of your chromatograms. So there might be a co-eluting peak that shares a mass in one sample that will get dropped as the quantitation mass, and a different mass will be chosen so that you have a clean quantitation across all of your samples. And it's super important that your peaks are quantified the same way across all of your samples if you want to compare one sample to another. So once we have aligned our peak table, then again, we're often going into one of these two global workflows, either biomarker identification or to look at global patterns. And what I'm going to focus on is looking at pattern recognition in our samples. There's often obstacles in our way for both of these workflows. We're often dealing with missing data. We're often dealing with sparse matrices, so really rare features that only show up in a subset of our samples. Sometimes we have very high intra-class variance where every sample is very different from each other. And then particularly for machine learning, this is especially problematic that because of the resolution of two-dimensional gas chromatography and metabolomics in general, we're often dealing with many, many more variables than observations. I'm not going to address any of those problems today. We've done some work and published some studies dealing with these various issues along the way. But what I'm going to focus on is once you've dealt with those problems, and you've dealt with missing data in your sparse matrices and your variables and observations and balances, then now you want to interpret your samples. What is all this data telling you? And really thinking about the fact that the goal of these analyses is to represent all of your data in one graphic. It's not very informative or helpful for us to look at each sample in isolation. It's impossible to write a paper talking about that. You want to paint broad pictures. So rather than looking at each chromatogram, which is represented by each one of these different puzzle pieces, when you're able to put them together into the big picture, you start to see how all of this information fits together, and it helps you to draw the larger conclusions. So we've already seen, I'm not going to belabor principal components analysis. We've already seen some examples of that today, and it's starting to get baked into software packages. So often we don't have to think a lot about it. We just push a button and PCAs are generated. What I want to talk about actually is how not only they can help us try to interpret our data sets, but it can also help us identify problems in our data sets. So we perform PCA at every single step of our data processing and analysis, when we're replacing our missing values, when we've just aligned our raw chromatograms, so that we can get a sense of the overall data structure and correct any problems that might be there. So here's a data set where we were looking for, we had two different classes of samples. And we were super excited when we first did a PCA of our data, and we're like, oh, look at these beautiful separations between our two sample classes. The separation had nothing to do with sample classes. It had to do with what day we did the analysis. And this wasn't even instrumental variation. We have our pooled samples down on the left-hand side of that PCA. What it was different batches of media that we were using to grow our cultures, the blue batch and the red batch. So we needed to correct this before we could go on to actually understand our system and interpret something meaningful from the data. But I also want to highlight that the principal components analysis, again, this is often our go-to. Very frequently, it is showing us less than half of the information contained in our overall sample set. And therefore, it might not be giving you the full picture of all the problems or all of the beautiful data structure that exists in your data. And so we're often turning to clustering analyses in order to actually look at the entire data set all at once. And we're often using some form of hierarchical clustering. So as I said, sometimes PCA shows you less than 50% of your data if you're just looking at PC1 and PC2. And so what I'm going to highlight is the ways that we combine the 3 C's, correlation, clustering, and color, in order to be able to get some global analyses of our entire data set in a single picture. First, some definitions. Correlation is a measure of similarity or dissimilarity between two or more things. And oftentimes when we are using some form of software to generate a clustering analysis, a hierarchical clustering, for instance, it's using one correlation method as the default. But there are many, many methods that you can use. I've only listed a few here. Pearson and Euclidean are our top 2 that we go to. There's Kendall, there's Spearman, there's Manhattan, and so on and so on. Bray Curtis. There are many options out there. So you might want to dig beyond the default. And let me give you an example of how we can use correlation analysis. So this is a mouse study where we looked at the lavage fluid from the mice. And we analyzed, they were infected with 2 different species of fungus, Coccidioides immitis, Coccidioides possidacei, and then we had some controls. We analyzed the lavage fluid, and we performed gas chromatography on it to look at the VOCs from that lavage fluid, and we also performed a cytokine panel. And one of our questions was, do any of the VOCs that we see in this system correlate to immune response? So we wanted to perform a correlation analysis. We, again, the baseline analysis, the default that we often end up using is Pearson correlation, which is just the linear correlation that we're all used to thinking of. And we had 16 samples across all the mice to calculate the correlations between any 2 features, whether it was a cytokine or whether it was a volatile. They could be correlating within volatiles, within cytokines, or between the 2 data sets. And with correlations, we're looking for either a perfect positive correlation, which would give you a 1, a perfect negative correlation, which gives you a negative 1, and then 0 is that there's no correlation. So here's a table, you're not meant to see these numbers, where we've calculated the correlations between each individual volatile and each individual immune marker. And along the diagonal from the top left to the bottom right is all ones because those are correlations to the exact same sample. We can't interpret tables that look like this. So if we turn these numbers into colors, we can start making sense of what these correlations are showing us, and we've turned this into heat maps. And so what we're seeing here on the top row is an immune marker, which is labeled at the top row, and how it correlates to every other marker that we measured. Either positively correlated in blue or negatively correlated in red or no correlation in white. So globally, we can start to see that all the immune markers seem to be somewhat positively correlated to each other. The volatile markers, there's a real checkerboard in there. There's positive and negative correlations across the volatiles and then in between the immune markers and volatiles. But this is still hard to interpret. We have to go line by line to understand the correlations within these sample types, within these feature types. So that's where clustering comes in. We first calculate the correlations, and then we cluster the markers by how they're correlated, whether they're positive, negative, or no correlation. Again, in your softwares, there are defaults for how these clusters get done. It's often something like complete, or maximum is the other name for complete. Maybe the default is average, but there's other ways to set up these linkages, and you should explore beyond the default. So here we've applied a Pearson correlation, as I said before, with average linkage. And now we can start to see global patterns finally of which immune markers and volatiles are positively correlated to each other, what they are now also negatively correlated with, and we start to see these neighborhoods of correlation across this heat map. And as I pointed out, one size does not fit all. You should go beyond the default, and I'll show you a data set and how much the clustering and the correlation can really impact that. So here's a sample set where we have the VOCs that are all in columns, and then we have samples in rows, and there are just 6 different sample types here indicated by shapes. And what we're expecting to see is that the shapes of the same type should be clustering together in this hierarchical clustering analysis. The default on this is Euclidean clustering in the columns, Euclidean clustering in the rows, and the complete linkage for each of these data sets. And we can see that the clustering is not awesome. But when we start playing around with different correlations and different clustering methods in rows and in columns, we start to see new patterns emerge. And finally, by using Pearson correlation and complete linkage on the rows, but sticking with the Euclidean and complete linkage in the columns, we're finally seeing clustering by our replicate samples, which is the minimum that we're expecting to see. And we also start to see patterns of VOCs that are generating these clusters within our samples. So think of it like tangrams. Try different combinations to generate a picture of your data that's easy to interpret for what you're expecting to see for your sample analysis. And then I want to make 2 pitches about color. Please be aware that a substantial portion of our population does not have perfect color acuity. If you are a person who does have good color acuity, you should be checking your graphics before you display them in publications or in presentations to make sure that they are interpretable by everyone. So you can see in this heat map, if you have high color acuity, that the way those symbols look on the left, whether you have one type of color blindness or another, can look really different, and sometimes you can't distinguish them. So make sure that you're checking these using different shapes when needed to make sure that they are color accessible. And then lastly, I just want to make a pitch for grayscale. Sometimes grayscale tells you the cleanest story and allows you to tell that story without a lot of visual interference. So that's it. I want to acknowledge the people in my lab who generated these data, Trenton Davis, Emily Higgins Kepler, and Daniela Gutierrez Munoz, and the collaborators who helped generate the data and the funding that helped generate that data. And then a lot of what I talked about, it was published in an LCGC article, and that's the QR code if you would like to access that. Thanks.
All right, questions for Heather? Going once, going twice. Well, I'd like to thank all of our presenters one more time. All right, and that is a wrap. I do want to make one announcement that for those of you that were interested in the automated derivatization, during the Tuesday lunch session, there's going to be vendor presentations, and I think we're going to plan on showing a short video that didn't work in Dave's talk. 4:25 is the time that will be. All right. Thanks again, everyone.